Welcome back to MLT 130. This lecture is going to be on laboratory instrumentation. First off, let's review the methodology of spectrophotometry. This methodology is used to measure how much chemical substance is absorbed by light. It's measuring the intensity of light as the beam of light is passed through a solution. And we've talked about this not only in clinical chemistry, but we've talked about it in hematology for things like hemoglobin. And we've also talked about spectrophotometry in laboratory calculations. So now let's remind ourselves some properties of light when we're talking about spectrophotometry. Electromagnetic radiation is photons of light traveling in waves. The frequency of a wave is inversely proportional to the wavelength. The wavelength is the distance between peaks in each wave. The entire electromagnetic spectrum includes things like gamma rays, X-rays, microwaves, UVs, and also visible light. And remember, when we're talking about visible light, we're talking about Roy G. Viv. So, like red, orange, yellow, green, blue, and violet. So let's focus on visible light for just a minute. Wavelengths typically used by our chemical or chemistry analyzers in chemistry are using visible light in that spectrum of 400 to 70 nanometers in that Roy G. Viv range. Chemical analyzers use spectrophotometric methods for many tests. Some may use UV methods or UV light, but the majority of the tests are visible light. Polychromatic is a light description of visible light, and it means many colors or having many colors. Poly meaning many, chromatic meaning colors. When we're talking about spectrophotometry, we always need to remember Beer's law. Beer's law describes the relationship between absorption of light of a solution and the concentration of that solution. The concentration of a solution is directly proportional to the amount of light absorbed by that solution. The percent transmittance is how much was transmitted through the sample. Remember, the light is shown directly at the sample, and we're trying to see how much light was absorbed and how much light actually made it through to determine the concentration of the sample. And you can look on page 84 in your textbook, figure 4-3, to see an example. And of course, we talked about Beer's law in hematology when we were talking about the hemoglobin measurement. So now that we've been talking about percent transmittance, let's have a trivia question. If all light is absorbed by the sample, what then would be the percent transmittance? The answer would be 0% transmittance. It was all absorbed by the sample. So none of the light was able to pass through the sample. This means the sample was very high or exceeds linearity. So at this point, we would have to do a dilution to measure, because we don't have any light that's passing through, so we don't have any way to knowing the concentration of the solution, because we don't have any transmittance. Everything was absorbed by the solution as far as light transmittance. Something we also have to consider or remember that we need when we're talking about a spectrophotometer or spectrophotometer results is a calibration curve. Results from a spectrophotometer are based on a calibration curve. Remember, we have to run a blank, and a blank is usually just consisting of the reagent itself, no specimen or no standard. And you run it first, and it's set at 100% transmittance. The calibration curve is usually at least 5 calibration curve points, and the concentrations are made up of known standards that are used to set up the curve, and it should be in a linear pattern. Just to remind us what a calibration curve looks like, I'll do my best to draw one down here, free-handed. So here is our curve. 0 would be the origin. I'm going to go up here and change colors to draw our standards to plot them. to create our curve. This would be our first standard, second standard, third standard, fourth, and again these are going to be in a linear pattern. Fifth standard. Usually have at least 5. Let me switch back colors. And again, these should be linear. Do my best to draw a straight line. And this is going to be absorption over here on this side and concentration down here. And whatever the reading is that comes off your spectrophotometer is going to be your absorbance. And you're going to use this, run it over to your standard curve, run it down, and that's going to give you your concentration for your solution of your unknown. Go over some of the parts that are included in the spectrophotometer. As far as the light source, it can be a tungsten or a tungsten halogen lamp, are most common for visible light. A deuterium lamp is usually used for UV light. There's the monochromator, which is an optical device that can transmit a narrow band of wavelengths of light, and it isolates the wavelengths required for the testing. There's the sample holder, which is usually a cuvette. They must be transparent and free of scratches or dirt in order for the light to transmit to the sample or through the sample. There's the photodetector, which converts transmitted radiant light into equivalent electrical signal. And then there's the readout device, or the computer, which can also be referred to as the LIS system. Of course, quality assurance is very important to a spectrophotometer. Typically, on most chemistry analyzers, during the daily startup, you will run quality assurance parameters to ensure that the spectrophotometric method of the analyzer is working properly. Wavelength accuracy implies the photometer is measuring at the wavelength selected. Stray light is any light that impedes on the detector that does not originate from the light source and can have a significant impact on the measure of the absorbance of a solution. And then linearity is the ability of a photometric system to yield a linear relationship between the radiant power incident and its detector and concentration. We need to know about a specialized type of spectrophotometry. It's called atomic absorption spectrophotometry. Its principle is a ground state atoms absorb light by defined wavelengths. It's used mostly to analyze metals such as lead, iron, magnesium, zinc, copper, and lithium in body fluids. It does follow Beer's law. The amount of EMR, or electromagnetic radiation, is directly proportional to the concentration of the metal in that solution. Light source is a halo cathode lamp. The key difference between the atomic absorption spectrophotometry and regular spectrophotometry is that atomic absorption uses a flame or atomizer. So an example containing metal is aspirated into a flame. Next, we need to talk about fluorometry. It's very sensitive, specific, and costly. The instrument that we use is known as a fluorimeter or illuminator, and it uses UV light. So let's talk about the principle of this methodology. Molecules absorb light and reach a high energy level called an excited state. Certain molecules then lose this excess energy, returning to a ground state. And in the release is a photon of light called fluorescence. Each fluorescent may have a characteristic excitation and emission wavelength, usually in the UV region of light. So there's some kind of fluorescent that's going to happen during that going from excited back down to the ground state. So what are the advantages and disadvantages of fluorescent testing? Advantages is that it's 1,000 times more sensitive than absorption techniques and is more sensitive. The limitations is it's sensitive to pH, also temperatures. It's susceptible to quenching, which is any process during the test that can decrease the amount of fluorescence from the loss of energy due to solutes or solvents. Another methodology we can use in clinical chemistry is chemiluminescence, and this is where there's an emission of light as a product from a chemical reaction. We could also use light scatter technologies in clinical chemistry. It's good for measuring concentrations of particles too big for spectrophotometric methods. Nephelometry is a measure of light scattered by small particles in a solution. The amount of scatter is directly proportional to the number and size of the particles in the solution. And then there's turbimetry, which is the measure of the amount of light blocked by suspension of particles. It's less sensitive than nephelometry. Another methodology is potentiometry. The technique is used to determine the concentration of a substance in a solution using an electrochemical cell that consists of two half cells, where the potential difference between an indicator electrode and a reference electrode is measured. And we can use this when we're measuring electrolytes and pH usually. Now let's talk about some of the electrodes that can be used in potentiometry. There is a reference electrode, which is the electrode against which all other electrodes are measured. It is the electrode with a constant voltage. The most common type of reference is a Coulomb electrode. Then there's the ion selective electrode, or the ISE, that we talked about when we covered our electrolyte unit. This is an indicator electrode. It measures specific electrolytes such as sodium, potassium, chloride, and CO2. It is a measuring electrode. And then finally, pH and blood gas analyzers use a pH-sensitive glass electrode. Now let's switch gears a little bit and talk about some separation techniques that are used in clinical chemistry. Let's talk about electrophoresis. We've already talked about this in particular when we were talking about protein electrophoresis. It's a separation of charged particles in a liquid medium under the influence of an electrical field. You apply a voltage and you use a buffer solution, and zone electrophoresis is the type most commonly used in the clinical lab. Some of the different types of media that can be used for electrophoresis include paper, cellulose acetate, agarose gel, and polyacrylamide gel. Some of the common uses for electrophoresis in the clinical lab can include our hemoglobin electrophoresis, the serum protein electrophoresis I mentioned just a minute ago, and also our hemoglobin A1C. Another methodology for separation is chromatography. It's a separation technique where solutes in a sample are separated for identification based on physical differences that allow differential distribution between a mobile phase and a stationary phase. So they're separated based on polarity differences. The mobile phase may be an inert gas or liquid which carries the sample. For the stationary phase, there's a few different options. It may be a silica or gel bound to the surface. It may be a gel plate or plastic sheet, or it may be a silica or polymer coated, or it may even be bonded with a column. So again, the stationary phase, it has some different options. There's some different types of chromatography. Let's first talk about thin layer chromatography, also known as TLC. It's used in a clinical setting for the detection of drugs of abuse in urine or DAU. It's usually a confirmation method in conjunction with a immunoassay screening method. Is a type of planar chromatography. Planar meaning lying in a plane of. The stationary phase is on a planar support, and it's in a thin layer of absorbent material. The next type of chromatography is gas chromatography, or GC. It's a separation technique that uses a carrier gas to move through a stationary phase located within a column. The carrier gas must be inert, meaning generally unreactive. Examples of carrier gases include helium, hydrogen, and nitrogen. This methodology is used to separate compounds that are generally volatile. The last type of chromatography that we're going to talk about is high-performance liquid chromatography. It uses pressure for fast separation, and it's controlled by temperature. It can be used for separation and quantification of hemoglobins. Another thing that we need to discuss that we test a lot of in clinical chemistry is immunoassays. Immunoassays are testing methods used to detect and measure either antigens or antibodies in a sample. An antibody here can be referring to a hormone, a vitamin, or a drug found in a biological specimen, such as blood. Immunoassays, like I said, are used a lot in clinical chemistry, and they are very sensitive and specific methods. In our immunochemical reactions, the immunoassays involve binding an antigen with an antibody, so the antigen can be natural or synthetic. Our antibodies or immunoglobulins used are from the 5 classes: IgG, IgM, IgA, IgD, or IgE. And of course, our IgG are the most prominent class used as a reagent. Just a few things to review for immunoassay. Our antigen can be large and complex, usually having multiple binding sites. The epitope is the binding site of the antigen, and this should be reviewed. The hapten is a low molecular weight antigen that has only one epitope. The affinity of an antibody is measured using hapten. And of course, our antibodies has a functional domain where it binds to an antibody. Thus, immunoassays are techniques used to detect very small amounts of analytes, so functions as antigens, by reacting with an antibody. Often in immunoassay testing, labels are added to the testing because we want to be able to visualize the reaction better. So fluorescent or chemiluminescent labels or enzymes are even added just so we can see those reactions or those results better. Now let's talk about enzyme testing. Let's first do a little enzyme review. These are biological catalysts, and they're proteins. They can be complex. Some have isoenzymes. CK and LDH are isoenzymes. Enzymes have an active site where it binds to a substrate. Substrate is converted to a product, and the enzyme leaves the reaction unaltered. With enzymes, you need to be familiar with the Michaelis-Menten equation, and it's E plus S, where the E is representing enzyme, S is equaling substrate. So the enzyme plus the substrate equals the enzyme plus the substrate. You're going to end up with the P plus the E. The P is representing the product, and of course the E represents enzyme. The enzyme's going to come out unaltered. These enzymes are proteins, so they can be easily denatured. Enzymes can lose their activity due to pH, temperature, or concentration of ions in the surrounding medium. Some enzymes even require cofactors or coenzymes for full activity. We need to understand enzyme kinetics. The measurement of enzymes in samples is based on enzyme kinetics. We have to react the enzyme to measure it. First order, the rate of the reaction is directly proportional to the concentration of the substrate. The zero order is the chemical lab analyzer uses zero order of kinetics. The rate of the reaction is dependent on the amount of enzyme. A plateau is reached where the reaction rate is independent of the substrate concentration. So thinking about enzyme kinetics, in an assay, to measure an enzyme in serum, we must react it with a substrate. As it reacts with the substrate concentration, it tapers off and eventually plateaus. The reaction rate then becomes dependent of the amount of enzyme, independent of the substrate. Now it's called the zero order, and that's why the assay uses the zero order. Also, there's a couple of terms that you need to know and understand with enzyme kinetics. There's the lag phase, which occurs after initial mixing of the specimen and the reagent. And then there's the linear phase, which is where the equilibrium is reached when the reagent rate enters the linear phase, having reached the zero order of kinetics. The final thing that we need to talk about is the unit of measurement for enzymes. We're going to be using the International Unit, or IU. The amount of enzyme that catalyzes for transformation is 1 micromole of substrate per minute of reaction at 25 degrees Celsius. This concludes our lecture on laboratory instrumentation. Of course, if you have any questions, as always, don't hesitate to reach out, and I will see you guys in lab.